joined today by our respected speaker, Dr. B. Sekar, former director and presently the consultant for BCG Vaccine Laboratory, Guindy, Chennai, Tamil Nadu. I'll just go very briefly and quickly so that we have more time to listen to Sir. But Sir is qualified MBBS and MD microbiologist with an experience of 33 plus years. Sir was just sharing now with us before the introduction. Sir is currently working in BCG Vaccine Manufacturing Institute, which supplies this vaccine to UIP, that is the Universal Immunization Program of Government of India. Sir worked as director in Pasteur Institute of India, Coonoor, and established the GMP, just now Sir again mentioned, the facility, compliant facility for the production of DPT group of vaccines. Sir worked in leprosy, also, which is one more important and public health significant disease in India for more than 20 years, and carried WHO ICMR sponsored lab and field-based research projects in all major diseases like leprosy, tuberculosis, HIV, and other bacterial diseases, and contributed many scientific articles in conferences covering areas like diagnosis, drug resistance, molecular techniques for leprae, tuberculosis, enterobacteriaceae, and vaccinology. Sir also published 50 plus scientific articles in reputed peer-indexed journals, and Sir is also a guide for PhD scholars in microbiology of Bharatiya University, conducted many training programs and workshops in the field of microbiology for emerging next generation of scientists. Sir also is acting as peer reviewer for international and national scientific journals. Let me just list out few awards Sir has received. Lifetime Achievement Award conferred by Association of Prevention and Control of Rabies in 2018. Lifetime Achievement Award in Microbiology by Indian Association of Applied Microbiologists in 2020. And now we are talking almost 15 years later. Belgaum, Mumbai Award for Best Microbacteriology Paper in 33rd National Conference of Indian Association of Medical Microbiologists. Also received Indra Vasudevan Award for Best Research Paper in Indian Association of Biomedical Scientists way back in 1987. Sir holds a couple of memberships in scientific societies as a life member for Indian Academy of Vaccinology, Immunobiology, Association for Prevention and Control of Rabies, Indian Association of Medical Microbiologists, Indian Association of Leprologists, Indian Association of Biomedical Scientists. With this very brief introduction for an illustrious career spanning almost 35 years, we request Dr. B. Sekar, our esteemed guest, to go ahead with today's webinar session. Thank you so much, sir.
Thank you, Dr. Sucharita. It's a very good introduction. Thank you so much on that. And at the outset, I thank Dr. Bala, who initiated this proposal, and Dr. Kannan, who has taken it forward, and you, Dr. Sucharita, who has come forward to have me in this evening on this platform. Thank you very much. At the outset, I would like to say, and rather I understood that this audience is an amalgamation of course, they have a scientific background from different faculties like microbiology, biotechnology, biochemist, life sciences, and a few of them from other field also. So having that in mind, I want to keep my presentation very simple. The language itself quite simple. And at some time, I may go a little technical just to feed the quest of my biotechnologist and microbiologist. But that won't affect the course of my information, what I'm going to put across to this August audience. So I would like to say this piece of work which I'm going to present has already been has come out on as an article in Times of India on February 22nd. So some of you might have who have read that, maybe you feel in some parts there may be some repetition, but still there are certain new information which I have added in this presentation. With this, may I have the first slide, please? Am I audible?
Sir, we can hear you, and you can share the screen and share your PPT, sir.
I think I already shared to Dr. Kannan.
No.
Can you show it, Dr. Kannan, or should I share?
You have to share, sir. You have to share and you have to operate the PPT.
Okay.
Yeah,
Okay. I'll do it.
Yes, sir.
Can you see this?
Yes,
sir.
sir.
Yes, sir. You can put it in
Right.
the slideshow.
Yeah. Is it
Yes.
okay?
Yes.
Yes, sir.
Full screen is seen?
Yes, sir. Full screen is visible, sir.
Okay. Yeah. Yeah. As a topic has been already been announced, the 100-year journey of BCG vaccine from cow to COVID-19. Before going on to BCG vaccine per se, I would like to tell you something about what is the problem of tuberculosis as such. About 1/4 of the world's population estimated to be infected by Mycobacterium tuberculosis, particularly bacteria. And according to WHO, an estimated about 10 million people fell ill with TB, and about 1.5 million people died globally in 2020, despite the fact that it is a very much preventable and curable disease. India also reported more than 2.4 million TB cases in 2019, and it continues to have the largest share of the global burden. And our national TB elimination program is having an objective to meet the goal of ending TB epidemic by 2025, which is 5 years preceding the global target. And as part of the preventive measures, although many preventive measures are available as for TB, BCG vaccine is the primary one, and it has been administered at birth, soon after the birth of the child, and also maybe till 1 year of the age. With around 160 million people vaccinated every year, BCG is the most widely used and safest vaccine in the world. Yeah. Next comes the history of BCG vaccine. How this BCG vaccine has come into play. As I said, it is a century-old vaccine. In 1901, it all started with Edmund Nocard. You may be knowing nocardiosis, a disease, a fungal disease. He has isolated Mycobacterium bovis, a bacteria from mastitis of a cow at Pasteur Institute of Paris. And this gentleman has transferred this isolate to two great French scientists, Albert Calmetti and Camille Guerin, who subcultured this vaccine for about 13 years. Interestingly, they used ox bile because they were encountering clumping of these organisms in the liquid broth, which they wanted to avoid, and they added ox bile to that. That ox bile addition has done some wonder. After 231 passages, they found out that the strain failed to produce progressive TB in the animals whichever have been vaccinated, have been injected with, and also exhibited a strong immunogenicity in these animals. Thus, the Bacillus calmette-guerin, BCG, which was named after the inventors, was born, and that is proved to be the approved vaccine for the human tuberculosis. This is how the Bacillus calmette-guerin BCG vaccine was born. And subsequent to that, from 1921 to 1924, a French veterinarian and also a microbiologist, Benjamin Ville-Hallet, administered this vaccine to more than 660 infants. This is what available in the literature. Orally, he was giving orally, and he found out no side effect among the children. The children did not develop any sign of TB. Later, realizing the fact that it has to go for a mass production, Pasteur Institute of Lille started producing in a mass level of this BCG vaccine. Of course, although originally it was administered orally, the current recommendation, as you all know, it is intradermal into the skin. What happened actually? The M. bovis, how they become a pathogenic mycobacterium, how it has become attenuated and become non-pathogenic. And later molecular evaluation has revealed what has happened between 1908 and 1921 when the serial passage was done by Calmetti and Guerin. During this passage, there are about 38 open reading frames have been lost. And among them, RD1, region of difference 1, which is a very important gene, has been deleted. This RD1, which is a 9.5 kilobase length, and it comprises about 9 genes, it also includes very potent genes encoding ESAT6 and CFP10. And both these proteins are highly T-cell antigenic targets and are also essential for the virulence of the M. tuberculosis. And deletion of this R1 is a key mechanism for the BCG attenuation, wherein the pathogenic MOVs has become non-pathogenic, and it plays a major role. I mean, this RD1, which since it plays a major role in the virulence, it has been deleted, and thereby the attenuation has occurred. And passing through these 231 passages, which led to the RD1 loss, also associated with other mutations, which can be a deletion, duplication, or single nucleotide polymorphism also. So with this understanding, let us move further. And from Pasteur of Paris, BCG strain was distributed all over the world since 1924, because it was felt that each country has to establish their own vaccine production facility, and it was further propagated, and local vaccine production commenced in each country. And as you all know, the freeze drying facility was not available in those days. So they have to keep these bacteria viable by subsequent propagation and subculture only. This propagation of BCG led to diversification to number of genetically distinct substrains called daughter strains due to deletion of certain genes. And mostly the deletion of RD1, I mean the region of differences, based on this deletion of this RD region of differences, two main groups of daughter strains have evolved. The first group, which is called as early strains, which evolved from 1921 to 1927, they include BCG. These are named after the country in which this propagation has been done, and this kind of changes, the genetic changes have been observed. So like in the early strains, 5 strains evolved between 1921 and 1922. They are BCG Russia, Moria, Japan, Sweden, and Birkog. And they had the deletion of RD1, also RD3, and also another gene, MB2377C. Later, when the propagation was going on in other countries from 1931 to 1961, the late strains evolved. They include Danish, Frappier, Coonoor, Ice, Mexico, China, Phipps, and Pasteur. And these groups, in addition to the deletion what we saw in the earlier strains, they also had the deletion of RD2, which contained the gene MPB64. And this antigenic, the gene encoding the antigenic protein MPB64 got deleted in the late strains. But whatever it is, although many strains have evolved during this process, there was no significant difference in the efficacy have been reported. There was no change in the efficacy of these strains, and all work same in different countries. And this is a schematic diagram which tries to tell you what has happened. See what has happened between 1908 and 1921, the attenuation of the MOVs into BCG carried out by Calmette and Gurin. From 91 to 1927, the early strains have evolved, which are called Russia, Moria, Japan, Sweden, and Birkog. Later, from 1931 to 1961, the late strains have evolved. Although we are talking about 2022 strains, but as of now, only 5 strains are being used worldwide for the vaccine production. They are Russia, Tokyo, Danish. Danish is the one what we use in our laboratory, Danish 1331, and Pasteur. These are the vaccines, the strains which are being used presently in the vaccine industry. Then after how this vaccine has been introduced in the immunization program. By late 1940s, several studies provided evidence for use of BCG in the protection against tuberculosis. And also, tuberculosis had emerged a major concern during the period of World War II. And BCG was encouraged by UNICEF in association with WHO and Red Cross Society. And in concurrence with that, in May 1948, Government of India has decided to introduce BCG vaccination on a limited scale under strict supervision as a measure to control the disease. Accordingly, a BCG vaccine laboratory at the King Institute, Guindy, Madras, was established in 1948. This is the institute I am working now. This institute now comes under the Directorate General of Health Services, Ministry of Health and Family Welfare, Government of India. And it has established a GMP facility also and started supplying vaccine to the universal immunization program. Initially, the work on BCG had started as a pilot project only in 2 centers, but by 1949, realizing the need of it, it has been extended to schools in almost all the states in India. Efficacy of BCG against tuberculosis. BCG, as we all know, as I said now, it has been included in the childhood vaccination program in around 180 countries in the world. And it is the most widely used vaccine in the world, as I said earlier. It is shown that in countries with high incidence of TB, BCG vaccination reduces the risk of meningeal tuberculosis and miliary tuberculosis, brain tuberculosis and disseminated tuberculosis by about 85% and pulmonary TB by 50% in infants. But however, this efficacy varies in adolescents and adults, ranging from about 0% in Chengalpet trial in South India, and which was carried out sometimes in 1960s or so. And in UK, in Medical Research Council trial, they showed efficacy about 80%. So there was a wide variation in the efficacy of BCG shown in different trials done amongst the adolescents and the adults. What is the reason for this variation? A meta-analysis, which was published in 2014, which did a systematic review of randomized control trials. The authors have examined association of trial settings and design with BCG efficacy against pulmonary and miliary or meningeal TB by systematic review, meta-analysis, and meta-regression. They identified about 18 trials reporting pulmonary TB and about 6 reporting miliary or meningeal TB. The review indicated that efficacy in pulmonary TB varied according to 3 characteristics. Very interesting. 1, protection appeared greatest in children who are stringently tuberculin tested. Tuberculin test is a skin test which is a diagnostic test which is done to determine whether the subject has been exposed to the TB bacteria or TB-like environmental bacteria. If suppose a person is showing the skin test positivity, he may be exposed to the TB bacteria or might be due to the environmental mycobacteria, which is similar to the TB bacteria. So the protection appeared greatest in children stringently tuberculin tested to exclude prior infection with the TB or prior sensitization with the environmental mycobacteria. But protection was weaker in children not stringently tested, which means the protection appeared to be more among children who have not been infected earlier with MTB or who have not been exposed to environmental mycobacteria. This is what this finding says. The second character is that protection was higher in trials further from incubator, where environmental mycobacteria are less and with lower risk of diagnostic detection bias. And it was shown that greater in trials conducted at the latitude farthest from the incubator, that is more than 40 degree latitude, it was absent or low in trials closer to the incubator, that is less than 20 degree. Again, the influence is due to the environmental mycobacteria. And thirdly, protection was higher when BCG was given in infancy at school age. And as I told earlier, this vaccine is effective only when it is given during soon after the birth or before they go for the school age. And no consistent evidence of protection in trials, including participants older than school age. So the older the children, the efficacy was found to be less. Of course, different BCG strains were put into, they were studied in different trials, but there was no difference as far as the difference in the BCG strains are concerned. They don't play any role in in determining the efficacy of the vaccine prepared out of it. And of course, as we saw earlier, protection against the other form of tuberculosis, like meningeal or miliary tuberculosis, was always high in infants and children, again, who were stringently tuberculin tested. So these are the salient findings comes out of this meta-analysis and the systematic review. And further, the review essentially reveals that, as I said, BCG vaccine is found to be effective in preventing pulmonary TB when administered in infants or young children not exposed to prior to TB or TB bacteria similar to bacteria which is present in the environment. And concurring this finding, a recent immunogenicity study also suggests that exposure to non-tuberculous microbacterial antigen could also block BCG vaccination from offering protection when infection precedes the vaccination. Yeah. Okay, what is the role of BCG other than tuberculosis? Apart from tuberculosis, this is one unique vaccine, as you should know, that we all come across a vaccine which are all target-oriented. If we use hepatitis vaccine, this can prevent only the hepatitis B, prevent hepatitis B infection. If you use meningococcal vaccine, it can prevent only the infection due to the meningococcus. But the BCG, apart from tuberculosis, has been shown to be useful in prevention and control of many diseases. This finding has started as early as 1927. A Swedish physician called Nastland reported threefold lower mortality in BCG vaccinated infants during the first year of their life, in their infant life, primarily due to the infection unrelated tuberculosis, that is due to neonatal sepsis or respiratory tract infection, which means BCG, apart from the protection against tuberculosis, also offered protection against the other infection causing sepsis and the respiratory tract infection. Further, studies conducted in 2005 and 2017 in countries like West Africa, India, and Haiti have shown reduced childhood mortality and hospitalization rates attributable to other respiratory infections independent of its effect on the tuberculosis. Even a recent study published in 2018 in adolescents in South Africa also reported 70% reduction in the respiratory tract infection by the BCG vaccinations. And also studies conducted in 2005 in Japan also shown even in elderly people, BCG protected respiratory infection probably due to the viral origin. Thus, BCG vaccination now known to be resulting in immunity offering protection to unrelated pathogens, and this kind of immunity is called as the heterologous immunity. And to understand this heterologous immunity, we should further understand what is called as the trained immunity. Many studies showed strong effect of BCG on the innate immune component of the host defense. As you all know, the immune response, primarily the early reaction is always innate immune response, and the later one is the acquired immune response. Innate immune response is a non-specific immune response, and the acquired immune response are the specific immune response mediated by the T and B cells. So BCG has got a strong effect on this innate immune component of the host defense, and it is initiated by PAMS. PAMS is called as pathogen-associated molecular patterns of BCG component. It binds to the host pattern recognition receptor, PRRs. BCG produces several PAMs recognized by the host PRRs like toll-like receptors, toll-like receptor 2, 4, 9, which induce a signaling pathway regulating the production of pro-inflammatory cytokines like interleukin beta 1, tumor necrosis factor, interleukin 6. And these are accompanied by transcriptional, epigenetic, and metabolic reprogramming of the myeloid series of the hosts in the BCG vaccinated individuals. And it was further shown these epigenetic changes are manifested as a chemical modifications. These epigenetic changes means they don't affect the codons of the DNA, but it is happening away from the codons, which is primarily due to a methylation or acetylation of histone, which results in the easier transcription of the gene important for antimicrobial response or their improved cell function. And in addition, metabolic reprogramming also happens, which leads to selective accumulation or depletion of certain metabolites that regulates this process because of their cofactor function for several classes of enzymes that mediate the epigenetic changes. BCG thus epigenetically train the innate immune cells like monocytes or natural killer cells to an enhanced host defense, and which is termed as the trained immunity, which is a unique character of the BCG vaccine. And this trained immune memory is non-specific, as I said earlier, because it mediates the innate immune response and protects against unrelated secondary infections independent of the specific immune response mediated by the T and B cell responses. And this is a schematic picture which can show you. I don't know whether you are able to see that. I'm not able to see. Yeah, this trained immunity of the BCG vaccination has shown to protection against multiple viral pathogens like RSV, influenza A, hepatitis, herpes simplex type 4, producing the epigenetic reprogramming of the monocytes, leading on to the protection of the pro-inflammatory cytokines. And this recognition of the pattern, that binding to the PRR, they induce this series of changes which happens in the cell, metabolic reprogramming, epigenetic changes, and as we saw, the histonic modification, leading on to a primary challenge vaccine, first response, initially low response, and a trained immune response of very high immune, secondary trained immune response. And this concept of trained immunity can be used as a tool for enhancing the population immunity during a pandemic. If you see lower down, in an emergence of an unknown pathogen, like as we saw with the SARS-CoV-2 now, trained immunity can induce initially through this innate immune augmentation, innate immune responses, the trained immunity by the time the specific antigen arrives. So this character of the BCG vaccination can be capitalized whenever we face an emergence of an unknown pathogen, which is attributed to the unique property of the BCG vaccine. As I said, BCG not only prevents the tuberculosis, it also helps in the other disease. It has been shown to play a very vital role in the prevention of leprosy. Widespread BCG coverage has significantly declined the cases of leprosy, and it was evident that BCG vaccinated population in high TB endemic settings had significant impact on the occurrence of the leprosy cases. Many studies have proved, as shown, and efficacy has shown in different trials. But on the whole, there is a good effect of BCG vaccine on the decline of the leprosy. And of course, it is equally effective both in the severe form of leprosy called lepromatous leprosy and the milder form of leprosy called tuberculoid form of leprosy. And given this fact that the efficacy of BCG vaccine against leprosy, countries like Brazil, Cuba, and Venezuela already recommended the use of BCG amongst the leprosy household contexts. And apart from that, BCG also plays a role in melanoma. Melanoma is a skin cancer. As an adjuvant immunotherapy, BCG has shown to be promising after surgical resection. Prior vaccination with BCG or vaccinia in the childhood had significant effect in case reduction compared to non-vaccinated groups. And of course, intralesional therapy of melanoma cancer with BCG has reported regression in about 62% in a study which I quoted there, Martin et al. In bladder cancer, BCG immunotherapy is known to be a standard therapy, especially the early form of bladder cancer called non-muscle invasive bladder cancer. It appears to be, it has been practiced as a standard therapy. In addition to other treatment, BCG immunotherapy intravesically, it has been administered, and it has shown about 70 to 75% complete response and also improved survival among those vaccinated with the BCG immunotherapy. Incidentally, BCG lab at Guindy was also earlier producing the cancer therapeutic vaccine. It will also very shortly come with that vaccine too. Then it also showed enhanced immune response to other vaccines. It shows enhanced responses with other unrelated vaccines by increasing their efficacies. It has been shown that BCG administered at birth augmented pneumococcal vaccine effects. It also enhanced the response to hepatitis B vaccine, increased the cytokines immune response to tetanus toxoid, and it also enhanced the immune response of the swine flu vaccine. The studies done in 2017 also showed there's a decreased severity of influenza infection. Yeah, this is one known fact which many studies have proved, have shown that BCG has got a good immune response in influenza infection. BCG and COVID-19. What is the role of BCG in COVID-19? If you recall, in the early phase of COVID pandemic, certain ecological studies suggested that countries or regions that mandate BCG vaccination have a lower number of infection and a reduced mortality of COVID-19. This is only an observational study. There is no definite proof of causality due to their inherent bias in these studies. So, and also based upon the hypothesis with the advantage of trained immunity of BCG might prevent SARS-CoV-2 infection or may reduce the severity of the COVID-19. To bridge the time until SARS-CoV-2 specific vaccine was available, more than 40 RCTs, randomized controlled trials, with approximately about 75,000 participants worldwide were initiated to evaluate BCG's potential in providing the non-specific protection through this trained immunity, in particular in people with high risk of for COVID-19, like health workers. However, the fast development of several COVID-19 specific vaccines, although it was good at that time, but it had got a bad effect on the BCG vaccine trials. The launch of this and also the launch of the national COVID-19 vaccination programs, it severely hampered the recruitment of COVID-19 naive and non-vaccinated participants for COVID-19 RCTs. Actually, it is not ethical to expect also, because when a specific antigen was available and shown to be effective, there was no place at the time for the BCG vaccine. Yeah, although it had its own potential. But meanwhile, a scoping review of 35 publications and trials to study the effect of BCG on respiratory complications caused by COVID-19 was published in December 2022 recently. This review concluded that the strongest evidence in favor of protective effect of BCG on respiratory complications of COVID-19 is currently provided by a BRIC trial, which is carried out in India by Singh et al. As the secondary outcome of the study. Singh has shown that BCG can reduce these complications in subjects with a chronic disease such as poorly controlled diabetes or chronic kidney, lung, or cardiovascular disease. But however, as of date, the remaining literature were mainly inconclusive and does not clearly either support or reject the theory. Meanwhile, ICMR initiated a study to evaluate the effectiveness of BCG vaccine in reducing the morbidity and mortality in the elderly individuals, more so in the COVID-19 hotspots in India. The study examined the impact of BCG vaccination which were all these elderly people were given 1 dose of BCG vaccination, and the study examined the impact of BCG vaccination on the frequency of T cell, B cell, monocytes, and dendritic subsets, as well as the total antibody level in a group of 60 to 80-year-old elderly people after 1 month, that is 1 month post-vaccination period. It has shown that BCG vaccination was associated with the enhanced innate as well as the adaptive memory cell subsets, as well as the total antibody levels in the elderly individuals, suggesting its potential in the utility in SARS-CoV-2 infection by enhancing the heterologous immunity, again through this trained immunity. But whether this translates to improved protective immunity to non-specific infection like SARS-CoV-2, it remains to be determined. Very recently, a Swiss study published in January 2023, which performed a systematic research for RCTs providing evidence about the non-specific effect after BCG vaccination. This study emphasized that although COVID-19 specific vaccines are available by now, there is a need for an assessment of BCG's special property of trained immunity effect and its potential as a bridge vaccination due to two main reasons. One, it is low cost. You'll be surprised to know the cost of BCG is about around 6 rupees. 1 dose costs about only 6 rupees, which you can never believe. It is low cost, wide availability, especially in developing countries, which often suffer from timely access to the specific vaccines and medical supplies. We have seen this in South African countries. They had the difficulty in getting the specific COVID vaccine on time, although other affluent countries, they could get it. Of course, we fortunately had the manufacturers of COVID-19 vaccine in India, so we could get the supply at the earliest and we got benefited. Number two, its potential to activate immune system against various pathogens like emerging vaccine-resistant SARS-CoV-2, immune escape mutants, or novel pathogens in future endemic. This what they strongly emphasize, that BCG, because with the property of trained immunity, has got a role in future also. Thus, BCG-mediated trained immunity may further reduce microbial infection also, because it through its heterologous immunity, they control the other microbial infections, and thereby lessening the drug prescription and the development of microbial resistance, because the irrational drug prescriptions leads to antimicrobial resistance. We all know about it. So this is what they indicate. So when it controls the microbial infection, the drug prescription will also be reduced, and thereby the antimicrobial resistance will also be reduced, and also may reduce the risk of all-cause mortality. This is what this study has emphasized, published in January 2023. A word about the recombinant BCG vaccine. So far, I was talking to you about the classical, traditional, live attenuated BCG vaccine, which we have been practicing all along. Recently, a vaccine called VPM1002, which is a genetically modified recombinant BCG vaccine derived from Microbiology bovis BCG subtype Paraguay. And in 1912, Vaccine Project Management, a German German organization, began producing VPM1002. This vaccine candidate expresses listeriolysin and deficient in urease, and is shown to be having a better safety profile due to lack of RD2 genome segment, because they are derived from the Paraguay. Paraguay does not have RD2. Yeah. In this vaccine candidate, urease gene is replaced with listeriolysin O, encoding gene from Listeria monocytogenes. By reducing urease G, the neutral pH becomes acidified, and thereby the phagolysosome fusion is more effective. That is how it makes a change when compared with the live attenuated classical BCG vaccine. And this vaccine has currently passed phase 1 clinical trial, tested in a phase 2A randomized clinical trial in healthy South African newborns. The target indication for this vaccine are newborn to prevent TB or post-exposure immunization in adults to prevent TB recurrence. And BPM 1002 is currently being studied for efficacy and safety in a multicentric phase 3 clinical trials, including in infants, including in India. And with this, what could be the future perspective of BCG? As you all know by now, BCG doesn't restrict its role only with the prevention of tuberculosis. It also plays a vital role in the prevention of multiple viral respiratory infections. It also plays a role in the cancers like melanoma and bladder cancers. And we also shown that due to its trained immunity, it has got the potential even to play a role in the future pandemics. So with that, the use of BCG, the future perspective could be the use of BCG for a variety of vaccination purposes, both specific as well as the non-specific, which we have seen over this lecture, has opened the door for renewed enthusiasm to further explore the application of modern recombinant vaccine technology for future vaccine development and immunotherapy. Thank you for your patience listening. Thank you very much.
Thank you, sir. We request you to, that was a wonderful deep dive across the span of 100 plus years. And I personally was very thrilled to see from slide 1 to the future prospects. That is how science evolves. And it has been like, I just remembered my professor as a postgraduate student in MD taking such a elaborative sessions, but you have given us a very beautiful kind of view how this technology or how this virus going through 241 subcultures have been shown with this wonderful favorable genetic deletions, gene deletions, especially the RD1, as you have said. And later you told us about this pattern recognition receptors. And this looks like very interesting because I recently completed a book called Genomic Machine, and it clearly explains the entire history behind how the technologies have evolved. So before I open this forum for discussion, I just request all our participants to either use the hand sign icons. So if you want to interact with sir, please use that raise the hand option icon so that I will moderate the discussion, and so you can unmute yourself and interact with our professor today directly. Because before I ask my own questions, I request the other participants in this last 15 minutes, we have a very beautiful window of time to interact and clarify any insights or questions related to today's presentation. Wonderful. I'm seeing some hands rising up. And before that, I thank all the 50 plus participants who have logged in for today's session. You would have missed it, the other people, but we are going to put up the recorded session in our YouTube channel. We request all of you to visit our Global Biomedical Foundation's YouTube channel, where all the archived materials will be there. So let me proceed with our regular webinar participant, Hanifi. Please unmute yourself and interact with our esteemed speaker. Hanifi, can you hear?
Yeah, ma'am. Am I audible?
Yes, you are audible.
Dr. Sucharita,
Yes, sir.
you may request other audience to put these their mics on mute.
Yes, sir. I request all the participants to mute yourself, especially we are hearing signals from Maharaja Pandian, sir. Maharaja Pandian, sir, we request you to mute. Thank you so much for your immediate response. Yes, we request all other participants to mute so that the speaker will be able to interact adequately without any background disturbances. Thank you so much for your kind support. Hanifi, please unmute yourself, and if you want, you can switch on your video profile and interact with our speaker today.
Ma'am, good evening, ma'am, and good
Yes.
evening, sir.
Good evening.
First of all, I want to thank you, sir, B. Sekar, sir, for wonderful and nice presentation. I have two questions on this topic, sir. First is, you talk about VPM 102. It's like first vaccine, and that they are administered to the UDH. Sir, like, can you highlight that if there is a not case of Helicobacter pylori upon this VPM 122 administration?
Can you repeat it? Your voice is breaking. Can you
Yeah.
repeat it?
You're talking about
No,
VPN 1002. Can you be more specific with your question and a little bit louder? Come near to the speaker so that sir can listen very
okay.
clearly. Yes.
Am I audible now?
Ah, yes, you are audible now. Yes, clear also.
Sir, like my question is, upon registration PM102, there is a urea efficiency. So can you highlight what's the effect on Helicobacter pylori, the VPM 2 effect on Helicobacter pylori? If
Okay. See, I know something about VPM, what has happened with the attenuation of the classical BCG strain. I am not an expert to comment on Helicobacter. I have not worked on that. To repeat what I said, there is a recombinant changes which has been done in the classical BCG attenuated strain, wherein the urease C gene was deleted. Instead, listerolysine gene was inserted. What happens is when the urease is there, it produces a neutralized pH which prevents or which does not stabilize the phagolysosomes. As we all understand, when a pathogen enters, they form phagosome, and there is a lysosome, bag of enzymes within the cell. The phagocyte, the phagosome, and the lysosome has to join together to form phagolysosome. Only then the enzymes, which are digesting enzymes present in the lysosome, will act upon the pathogen and destroy. This is what conventionally should happen. But what happens is in the classical strain of BCG, this phagolysosome fusion is not complete because of the neutral pH which exists in the cell. But when you, after doing the mutation, deleting the urea gene and inserting the listerolysine, an acidic pH is formed. This acidic pH favors the fusion of phagolysosome, and thereby this bacteria can be killed easily. So this is what I was trying to explain to you. I'm sorry, I'm not an expert on Helicobacter pylori. I cannot comment on that. I hope I have satisfied.
That was a very great, clear explanation about that recombinant DNA vaccine. Now, Hanifi, you said you had one more question. You want to pose that?
yeah, ma'am. First of all, thanks for this first clarification. And my second question is like, tuberculosis bacteria form a biofilm over our lung surface and everywhere. So is the BCG vaccine has proper effect or on this biofilm? Because nowadays biofilm is very in the discussion, hot topic discussion.
Yeah.
So I want to know.
No, I have not come across any, personally, I have not come across any biofilm induction, control of biofilm production by microbial tuberculosis by the BCG vaccination. But overall, the action of the BCG vaccine, as I told you earlier, they act through the innate as well as the specific immune response. But what happens at the biofilm level, honestly, I don't know, Hanifi.
Thank you, sir.
Thank you so much for that very, very interesting, brilliant questions. You are always the star questioner of our webinar series, posing very interesting questions. Thank you for that.
Yeah, it was really interesting.
Yes.
Anyway, I think this provokes me to read about biofilm also. Thank you.
Yes, sir. That's why we want youngsters to attend our webinar so that they will stimulate us to read beyond our research and explore more.
Exactly.
Thank you so much.
Younger brains are brighter brains.
Very true, sir. Now, there is another hand raised by Sabah Manvili. If I'm wrong in pronouncing the name, kindly excuse me. You can unmute yourself, and if you are comfortable, you can video format, you can interact with our professor.
Thank you so much, ma'am. It's really a wonderful session. I was attending the last two sessions. I just came to know this webinar for the last two sessions. It's so wonderful. The arrangement made is also so wonderful. The session is also so excellent information we have heard. Actually, sir, this BCG, you said it is not freeze-dried or lyophilized yearly. How the stability has been maintained? Can I know that?
Yeah. See, as we all know, only in 1960s, the freeze-drying procedure has come into play. Before that, if at all you want to keep the, this is a live attenuated vaccine, you have to have the bacilli live, if at all you won't have the BCG effect. So it was all due to subculturing. As we do a subculture in the bacteriology, so stocks after stocks, this subculture was going on. That is the reason the repeated subculture has induced the genetic changes, deletions, SNPs, insertions, all those things happened. It is fortunate that even from the period of Calmette and Guerin, this deletion has resulted in the loss of the virulence factor RD1. So that is how the culture was preserved, rather propagated through repeated subcultures before the era of freeze drying. Yeah.
Now it is a freeze-dried product.
Now it is freeze dried. Now we follow what is called the seed lot procedure. We produce sublots from the master seed lots, and each lot goes for certain number of propagation. After that, it is not stopped, because we don't allow the propagation to continue, because this kind of genetic deletion and addition may happen. That is why it is stringently followed now. You won't see much of variation in the gene component of the currently used strain. We use a Danish 1331 strain since 1948. It has got a stabilized gene pattern because of the availability of the freeze drying and following a seed lot practice, manufacturing practice.
Okay. Sir, what is a normal stability, that is shelf life of a vaccine?
It is about 2 years after the production. After it is get filled up, it takes about 2 years. 2 years of expiry period you can expect.
So all condition like India is falling under zone 4, whether we are keeping in zone 4 stability conditions?
Exactly. There is something called VVM, Vaccine Vial Monitor. Is a very strict thing that we have to affix on each vial. This vaccine vial monitor, which monitors the temperature it gets exposed to. If it has been exposed to the advent temperature, the color of the monitor label will change. Even if it is within the expiry period, if the monitor color changes, you cannot use it. Such a stringency is being followed. Yes, in India.
Is it only a color change? It is a visualization or any other procedures like
No. You can normally
sterility, other things.
You
Content
don't
of the thing.
you don't need any device. Just by your naked eye, you can see. It's a label, just a sticker. It's a sticker which is sensitive.
Like color change after some period of time. You won't use it after some period of time. Color
changes if it is exposed to advent temperature. The required, the optimal temperature of storage is 2 to 8 degrees Celsius. If it crosses beyond 8 degrees Celsius, over the period of time, the color will change. That is the function of the VVM, vaccine vial monitors.
Okay. So, what about the difference between this control trial and observational studies, which I've seen in one slide?
I think Dr. Sucharita can tell you. I am not a community medicine.
Definitely, I can definitely help you, Sabha.
Yeah, please.
Observational studies are like you do not intervene with any intervention, whether it is a solution or any service or a product. You do not intervene. You just observe what is happening in that study population from which you have selected a sample, representative sample.
Yeah.
Whereas the randomized control trial is the gold standard for evaluating as Sir has said, efficacy, safety, efficiency, whether there are any side effects, serious adverse reactions, all these factors of a new technology or a new vaccine or candidate vaccine can only be undertaken if you have designed a randomized control trial, which is the gold standard of clinical experiences.
Yeah,
So
ask it.
yes, I would request you to read in detail because we have other hands showing
Yeah,
up. Right?
yeah, thank you. Thank you so much. Thank you so much, ma'am.
Yes.
Thank you so much, sir. Thank
The
you.
information will come from the heart's mouth.
Yeah,
Yeah.
thank you.
And now I
Thank you.
request Dr. T. Sekar to unmute yourself and interact with our professor. Dr. T. Sekar.
Yes, ma'am. Thank you, ma'am. Yeah. Thank you, sir, for your very good presentation among this BCG vaccines.
Thank you, my
And my
namesake.
doubt is, my doubt about it is, you said about the cross protection against the viral infections, specifically for the influenza and SARS, these kinds of infection.
Yeah.
Is there any specific mechanism against to these viral infections, or it is acting against the innate immunity like
Yes.
that?
Yeah. As you rightly asked, see, any vaccine will have a specific immune response induced action only. But the unique property of BCG, it shows the non-specific immune response also through what is elaborately explained as a trained immunity. This trained immunity influences the innate immune cells through different epigenetic changes and prepares these innate immune cells of the host for a pathogen which is not related to the target it is meant for. So that is how this BCG vaccine works against the multiple virus infections through this heterologous immunity. It's all due to the trained immunity, as I explained to you.
Yes, sir. Yeah, thank you, sir. I have another one more doubt on it.
Please.
You have said that the younger children are more immune when compared to the adolescent people.
Yes.
For this attenuated strain of BCG vaccine.
Yeah.
So, is there any specific cells which is responding to the antibody protection or cytokines or interleukin protection? And is there any specific mechanism involved in it, sir?
I doubt there is any change in the immune mechanism between the young children as well as the adolescents or adults. As I was telling you, the exposure to the MTB or to the environmental microbacteria. See, this vaccine is effective only in the naive individuals who have not been exposed to MTB or to other environmental microbacteria. But as the person grows, the chances of getting exposed to either to M. tuberculosis in an endemic country or to the environmental microbacteria are more. So the exposure to the mycobacterium tuberculosis or sensitization due to the environmental mycobacteria, they are the prime reason why the adults and adolescents or older children are not having the same protection as we see in the children and infants and newborns. That is the solid reason.
Yes, sir. Thank you, sir. Thank you so much, sir.
Welcome, Sekar. How are you?
Yeah, fine, sir. Fine, sir. We are happy to see you, sir. Yeah.
Yes. That's an excellent deep dive again, because it's very important for conferring cross immunity, as sir has explained, across variety of respiratory infections and other leprosy and all that. That's a wonderful question. Thank you, sir. Now, Dr. Sharavanan also can unmute. Yes, sir. Please.
Yes, madam. So thanks. It's another wonderful presentation.
Thank you, sir.
My question, sir. Usually in general evolutionary biology, especially the molecular evolution, the DNA is much stable than RNA, but TB is in DNA. But despite the stable genomic nature, how come can get too much of mutations in tubercle bacilli? Is this applicable for other bacterial strain as well, or only for TB? If it is only for TB, what could be the real mechanism behind this? Too much of mutation.
Yeah, this happens in all the bacteria which we know about, sir. Because although we say DNA is a very sturdy molecule compared with RNA, this process of subculturing repeated subculturing, they induce the genetical modification. That is the reason recently we are not allowed, even though we keep a controlled temperature to store all our isolate, we are not allowed to take the passage or subculture of one lot to more than certain level. Because this repeated subculture, because there is a change in the pH, change in the environment of the media, all those things can induce the genetic modification. It does happen. It does happen in other organisms also. And this BCG has undergone exclusively high number of subculture. As I told you, 231 passages. The grade 2 men, they spent their whole life on this. That was one reason why the RD1, luckily, fortunately, the RD1 got deleted and pathogenic MOVs has become a non-pathogenic. It does happen, sir. Even though we say that DNA is a very sturdy molecule.
Yeah, thanks, sir. But the question why I asked, because in generally we used to keep the subculturing of ATCC strain in the lab. So if we fix some kind of the threshold beyond that subculturing point, I think we should not do, because likely to create mutations, can also express phenotypically, because that is what we use as a standard, phenotypic characteristics to assess other, isolates in our clinical isolates of a lab.
Exactly.
So
sir.
yeah. So if we could come up with some set point beyond that level of subculturing, we should not go ATCC as well. So this is applicable for ATCC as well, right?
All the isolates, sir. Even ATCC, the manufacturer manual, if you carefully see, they'll tell you the optimal temperature it has to be stored and optimal subculture it has to go. It happens with any bacteria or even the viruses too. Yeah.
Yeah. Thank you so much, sir. Thank you so much.
Welcome, sir. Welcome,
Yes,
sir.
that's again one more interesting take on this unique presentation. Now I request a participant who is identified as iPhone. Unfortunately, no name is there. Sir, you can please proceed, iPhone participant, and interact with our professor.
I'm sorry, I thought my name is I'm Gopal from Malaysia.
Oh, wonderful, sir. Welcome.
Sekar, Dr. Sekar, how are you, sir?
Fine, sir. May I know your name?
Gopal.
Oh, Gopal, my friend. How are you?
Fine, sir.
Yeah, tell me, Dr. Gopal.
You said there's some role of your BCG vaccine to play in this COVID situation, correct?
Yeah, but there is no direct evidence so far because unfortunately, most of the trials got truncated. As I said earlier, because of the availability of the COVID vaccine, this study could not proceed further. Out of the known about 40 RCTs which were carried out, which were initiated rather in different countries like Australia, New Zealand, UK, US, but they could not proceed further for a very valid reason because specific vaccines were available and they didn't want to put the subjects into risk. And that could be the reason why most of the studies were truncated. Very few studies, as I showed in one of the scoping study, has come out with that. One trial has shown that it is effective against the protection of the respiratory complications. But by and large, if you see the character of BCG vaccine due to its trained immunity, it has got the potential, but more studies are required if it is possible. And in any other pandemic also, you can dominate this trained immunity characteristics of BCG before the arrival of the specific vaccine. That is strongly been advocated.
Thanks, sir. Thanks, sir. My further question is that we have some people who are not vaccinated with BCG at a young age.
Yeah.
What do you suggest for them to do? Should they get vaccinated or just leave it alone? Or,
sir, even though, as I said, majority of the population has been exposed to tuberculosis, only 10 to 15% suffer from disease, which means 80 to 85% of the people, they have some kind of immunity to resist the multiplication of the microbacteria, and thereby resulting the disease. So by and large, the population, especially in an endemic country like India, they have got a natural immunity. So don't trouble the trouble unless the trouble troubles you.
Third question is from Malaysian perspective.
Yeah, could be. The same may be true with Malaysia also.
Yes, sir.
Yeah.
Because as we know, BCG vaccine does not protect, does not have the efficacy in the adults. Of course, there are certain newer thoughts are coming. Revaccination of BCG. The children who have been earlier vaccinated, can they be given again one more vaccine? These are some studies coming up, and it may come. It may come. There are some strong evidence to show that revaccination helps in the better immune response against TB. Yeah.
If I'm allowed to ask one more question,
Please.
what is the best way to assess if an individual is got exposed to your tuberculosis?
The simplest is gamma interferon assay.
Okay.
That is one in vitro assay we can do. Nowadays, you remember the tuberculosis skin testing, which we were doing with the Monto test.
Yes, sir.
The Monto test, we used RT23 as a PPD. But now there is a different form of antigens are coming, CTAB. It's called a CTAB, which exclusively stimulate the ESAT and CFP10. So they are found to be more specific than the Monto. Monto is not a specific tuberculosis test. All it tells you, whether you have been exposed to MTB or to any other similar environmental bacteria. But this CTAB, which has been undergoing some trial, even in India, this trial is going on, phase 3 trials are going on. That will be more promising because it acts upon the ESAT and CFP10 specific for the M tuberculosis. That would tell you whether a person has been exposed to M tuberculosis or not. Certainly, it will have a greater implication than the RT23 FPPD.
Thanks a lot, sir. I'll catch up with you later.
Yeah,
Thanks a
we'll
lot.
talk.
Thank
later.
you. That
Thank
was
you for
again
joining.
wonderful insight.
Thank you,
Yeah.
Dr. Suchitra. Thank you.
Yes, sir. Thank you for joining us from Malaysia. We are happily thrilled with the international participants too. And Dr. Bala, you can unmute yourself.
Thank you. Thank you so much.
Thank
And
you.
excellent presentation, very informative.
I should thank you, Bala, for initiating it.
And not like that, sir. So everybody should be benefited out of this.
Yeah.
So that apart from my thankfulness to you for this presentation, so I thought of asking that other thing that when RD1 deletion is playing a major role in terms of making this BCG as a vaccine, excellent vaccine. Why are we not genetically modifying this and using it and continue to use live vaccine? Why can't we delete manually by genetic engineering and use it?
No, this kind of gene construct one could do. That is called gene knocking down or gene deleting. We can do, but it's already available. BCG is strain by virtue of the grade 2 men, their hard work, they already resulted in RD1 deletion, and it is already available. And of course, although we talk the efficacy of the BCG vaccine has got its own drawbacks also. That is the reason why we are coming out with the recombinant BCG vaccine, like as I said, VPM 1002. There are many VPMs, VPM 104, 101. There's so many VPM recombinant candidate vaccines have been tried by the vaccine project. This VPM of company of Germany. They try to find out whether these recombinant vaccines will be able to surpass what are the problems what we have with the BCG vaccine. Although we say it is highly effective, it is not effective in all the individuals. So these recombinant vaccines play a role. There are the phase 3 trials are going on elsewhere and in India also. Perhaps they may throw more light on the applicability of the VPM recombinant BCG vaccine. That may be the better way to look forward.
Yes, sir. The other thing is, so due to trained immunity, that it has got a lot of value apart from protecting against MTB, and so that you just listed out so many advantages in terms of BCG vaccination. Why are we not giving booster dose so that will improvise and even protection, and that will help a lot.
Yeah, there are some studies which supports the revaccination. That's what I told in response to one of the participants. Revaccinations is being thought now, and even India also, Ministry of Health is actively looking into the possibility of revaccination. So if that comes into the program, maybe initially it may come as some trial, and it may come as a program. Perhaps if it comes in the program, a TB control program, perhaps we may see the result. And whatever we anticipate, the trained immunity of BCG vaccine has got a very good role to play in preventing many viral infections. Even in pandemics, in the initial phase of pandemic, one can use the BCG vaccine because of the potential of the trained immunity inducing the innate immune response. Yeah.
Thank you, sir. Last question. And why are we not using this passage concept for other pathogens? Why that we go for the newer? Because even if you take SARS-CoV-2, and I don't think that I have seen anywhere this kind of effort has been tried.
You mean the restriction of passage you are talking about?
No, making it attenuated.
Okay. Attenuation is a very tricky process. Calmetic Gurin were lucky that even got deleted. But I don't think that attenuation comes so easily. Yeah, polio is an attenuated vaccine. Yeah. Sabin was lucky enough to come across polio type 1, type 2, type 3. Although we had a Salk injectable vaccine, still Sabin oral polio is still has got its potential. Now, for the endgame strategy, WHO is coming out with the killed Sabin vaccine. Sucharita may be knowing about it.
Yes, sir.
Yeah.
Yes.
Killed Sabin strains, not the Salk strains. They are planning to come out with the killed Sabin strains. That will be good. Yeah.
Thank you, sir. Thank you so much. Thank you,
Yes.
Sucharita madam and Dr. Karna.
Thank
No,
you,
sir,
Dr.
we are
Balu.
thankful. We are thankful for this wonderful session by Dr. Professor especially. And I have got 2 more questions in the chat box, sir. I will read out to you. Are
Please.
there any placebo controls included in the earlier vaccine trials? This was a question raised by Sushma Jadhav.
Very tricky question. No, what could have been was that the previous trials, they would have taken the BCG vaccinated as one cohort. And the other cohort, not intentionally placebo, those who have not been vaccinated, they would have taken as another cohort for comparison. This is how most of the RCTs they report, because at least in the earlier part of the period, the acceptance of the vaccine would not have been uniform throughout. So the vaccinated, by and large, who have accepted for vaccine could have been taken as a test cohort, and those who are not got vaccinated, not intentionally, those who are not got vaccinated by their own choice, might have been the control cohort.
Yes, sir. That is a wonderful interpretation, because as you correctly said, it was way back, all these trials were done. There could be some kind of
ethically it is not also correct,
And from Amudavalli, sir, is trained immunity and immunomodulation same concepts?
I think immunomodulation is not equal to trained immunity. The trained immunity, it acts more in the innate immune responses, unlike the immune modulations. And exclusively the BCG vaccination, as I've explained to you, this trained immunity enhances the innate immune cell responses through the natural killer cells and the other monocytes. So it differs. The trained immunity is different from the immunomodulations.
Wonderful. And there is earlier participant Sabha who is posing question in the chat box. What about the regulatory approval process for vaccines in India? You can give a very brief process, sir, because it may be elaborate for you to take time.
Rather, you are the right person to say. Anyway, to make it very brief,
Yes,
see,
sir.
when your vaccine is new, you have to undergo clinical trials, as what we have seen in the COVID vaccine. But COVID vaccine has taken a different route. As I said, UAA, emergency use authorization, that provision was made. They did restricted phase 3 or phase 2A clinical trial. Of course, phase 3 clinical trials are also done in a restricted sense. And of course, phase 3 trials very much necessary to prove the efficacy, safety of a vaccine. After doing that, you can come out with the vaccine. With this result, you can approach the regulatory authority. The regulatory authority has got their own way of scrutinizing the document, both for market authorization as well as for the production, because you should have a GMP facility, BSL-3 facility in case of the disease like SARS-CoV-2, and they will ensure whether your facilities are as per the standard expectations, and you have to apply. And after these processes, subject expert committee also you have to face and present your data, and subject expert committee will decide whether your vaccine can be marketed or not. And briefly, I'm talking to you about, not in depth. And with that, the commercial license will be offered to you. Before the commercial license, you have to do the trial batch production and consistency batch production. You have to show that the vaccine has got a consistency property throughout, and it has to get tested externally through an institute called Central Drug Testing Laboratory in Kasoli. They have to say that your vaccine is consistent in performance, and after that, they will issue the certificate of release. With that, you have to approach for the commercial license. Again, for commercial license, the inspection will go on, joint inspection, mostly by the central drug authorities as well as the state drug authority. If they are satisfied, then the commercial license will be offered to you. Then after the commercial license, you have to produce the vaccine. It's not a small journey. The journey is quite long.
Yes.
To market a vaccine, it takes 5 to 6, 7 years easily.
That's what sir was emphasizing. With that kind of backdrop, the emergence of SARS-CoV with the COVID-19 avatar, emergency use authorization has been kind of taken as a exceptional approach for bringing the pandemic to a halt. And with this kind of opportunities and technological revolutions, with the artificial intelligence, machine learning, big data supporting, and the entire infrastructure leveraged, still, as sir has mentioned, it is an arduous process. Deep pockets, big pharma, big institutions will be able to invest that kind of human as well as technological investments apart from money to go through this entire rigorous process.
Exactly.
That was again one more deep dive into the another window of vaccine regulatory procedures. Now, our participants have enormously thanked you, sir, for this wonderful, excellent, brilliant session.
Thank you very much. I'm very happy that I interacted with one of the very strong and intelligent audience.
Platform audience, yes, sir. Definitely.
They also gave me some homework to read something about. Sure, I will read, because learning is a continuous process. Yes. We all know that.
Probably we would request you to come back again and take us through one more session of your expertise as a consultant in this famous institution with historical legacy. With that, we thank all our participants for appreciating. Dr. Srinivasan has appreciated our weekly Zoom webinars. Thank you so much. And we thank all our international and national and regional participants for joining us today for this deeply intellectual high-end session. With that, we take your leave, thanking our professor for his valuable time. And we promise you to come back next week with a woman speaker, because we are doing the International Women's Day event on March 9th. So we will identify an illustrious woman speaker, and we request any of our participants to name recommendations or any of your suggestions, we take it forward and we will invite those professors. You are always welcome to suggest topics, speakers from your network so that we will invite them into our platform. With that, we thank our professor once again. Sir,
Yeah.
it was a very honoring time with you, and we wish you well, and we will see you soon.
Thank you. Thank you very much. Thank you very much for giving this opportunity and giving this interaction with the young and matured audience.
Yes.
And I should say that it had given me a chance to brush up my information and prepare this PowerPoint, although it was little, I made some additions also for this crowd.
Yes.
For
It
this
was so up to date as far as January 2023 also. You have given us updates, food for thought, and really going and keeping up pace with that. Thank you, sir.
Yeah.
Yes.
Thank you. Thank you all of you. Thank you
Yes.
very much for patient
Yes.
listening and for the interaction. Thank you.
Yes. We'll all see you next Thursday.
Sure.
Yes.
Sure.
Thank you. Thank you so much.
Thank you.
Have a great evening.
Great evening too.